the frequency of individuals which are homozygous of a different, of a allele, say big A, homozygous or allele small a, or heterozygous. And then you ask, what is the frequency of the A allele, where the frequency of the A allele will be twice the frequency of the homozygous, times the frequency of heterozygotes, and of course the complementary frequency for the other alleles. We are considering a simple case in which we only have two alleles. And then you find that if some conditions are satisfied that we are going to discuss, there is the frequency of the genotypes can be expressed by the frequency of the alleles themselves. So what are these assumptions? First of all, you have large populations. That means that you forget about the sampling fluctuations that we are going to discuss later. So fluctuations are neglected. It's purely deterministic. Second, neutral genotypes. It means that there is no selection effect over which alleles appear and whether on this. So all individuals have the same fitnesses, at least as far as this locus, this particular gene is concerned. And third, which is quite important, is mating is random. There is no individuals having one genotype to choose and mate with the similar or a different genotype. Then if these conditions are satisfied, you can find that within one generation, whatever the initial values of the frequencies of the genotypes were at the beginning, within one generation, you have that the frequency of the heterozygotes is equal to p squared, the frequency of the homozygous for A is p squared, the frequency of the homozygous for small a is q squared, where p and q are the quantities that we have evaluated in the first slide as a function of the initial values of these frequencies. And that means that in fact we can focus on the allele frequency instead of focusing on the genotype frequencies. I remind you, provided that these conditions are satisfied. And that, from this point of view, this simplify, I can draw, this is a nice way of representing the relation that I just wrote about. So here we have what is called a barycentric diagram. The idea is that if any point in this triangle describes the composition in genotype of a given population, and the distance from the vertices is, let's put it this way. Imagine that you put a weight on each vertex, which is proportional to the fraction of individuals with that genotype. And then you ask, what is the position of a center of mass of the triangle? So it will be some point which lies inside the triangle, and this point is determined by the frequencies. Of course, it goes closer, for instance, to AA if the frequency of big AA goes towards 1. And if we have, for instance, half homozygous for big A, half homozygous for small a, will be the center of mass will be somewhere here, and so on. Now imagine that you are given a certain frequency of the allele big A, let's call it P, and then all this line here is made of all the possible compositions of the population such that the frequency of allele big A is equal to P. It's all this vertical line. And then the Hardy-Weinberg equilibrium line is, you see, is this line here that is described as you change the value of P. And what you find is that you can start, for instance, from a population which has a composition P0, and within one generation, provided that these three conditions are satisfied, you end up at this point Q, which is the intersection of this vertical line with the Hardy-Weinberg equilibrium line that you find here. Now, this is just to say that we can forget about the genotype frequencies and we look at the allele frequency. And mutations happens, mutation is a way by which new alleles can be generated. Because what we know is that, for instance, in genes are described by sequences of nucleotides in DNA, and these nucleotides come in four different brands, and we are focusing on a very simple, on the very simple mechanism in which variability can be introduced in a given sequence. That is a situation in which, by which one nucleotide changes its type from the parent to the offspring. So sequence mutations are changes in the offspring DNA with respect to that of its parents. And the simplest one that we consider are called the point mutations, mutations of a nucleotide into a different nucleotide. So these point mutations can be classified according to their nature in two kinds. So one kind is that A goes to G or C goes to T. The reason is that these are purines and pyrimidines, two looped or one looped nucleotides, and the transitions keep the number of loops, keep the nature of the nucleotide at this level. So they are, and the transversions are changing purine in pyrimidine and vice versa. And of course, you have also other forms of mutations that are important that do not fit into this scheme, that is insertions or deletion of a short nucleotide sequence. You can even have major reshuffling, but I'm not going to discuss about major reshuffling at all. So we have this scheme, and you have, in this case, we have transitions here, transition here. So if we put, and then transfer all these other changes here are called transversions. And of course, what you expect in general. Okay, now this is about the nature of the mutation. What is the effect of mutation? Let us focus only on coding sequences, that is those stretches of DNA which will be translated into messenger RNA and then produce proteins. In this case, we know that the proteins are made by reading off the sequence of nucleotide and translated via the code into amino acids. So we can look at the effect of a point mutation. And the effect of point mutation, you can have either synonymous or silent mutations. Synonymous or silent mutations are the ones such that the mutated sequence produces the same amino acid in the eventual protein. So what you assume is that in general, these silent or synonymous mutations will have a weak effect. I wouldn't say it is far-fetched to say that they will have no effect, because there can be indirect effects that I'm not going to discuss, but it's a weak effect. On the other hand, you have a stronger effect, that is a situation in which you have that a codon, a triplet of a nucleotide, will change into a triplet in codon, which codes for a different amino acid, but is still an amino acid. In this case, what you end up is having a protein which has the same length, but in which one residue, one particular amino acid, is changed into a different one. And of course, this will have, in general, some effect on the protein itself. So we have, in this case, an important effect. And then we have the so-called nonsense mutations. That is a mutation in which you have a coding triplet, which is by mutation changed into a stop sequence. So what it ends up is that at the beginning you have a protein of some length, and then because of this mutation, the mutated sequence will code for a much shorter protein. And you can imagine that this has a much stronger effect than the previous ones. So you can classify mutations according to their effect in coding sequences. What I'm not absolutely discussing here, what happens when the mutations take place in the non-coding sequence. You can imagine this is much harder to characterize. And in fact, say, there are at the moment no general method to characterize the effects of mutations in the non-coding sequence. Moreover, indels with a length which is not a multiple of 3 produce a reading frame shift. That means a sequence that you end up having will have, starting from the point of the indel, whether it is an insertion or deletion, it doesn't change much.
What's an indel?
Indel is insertion or deletion of a certain subsequence. If this sequence has a length which is not a multiple of 3, then downstream, what you are reading the codons in a different frame, and therefore the whole sequence of the genetic code will be read completely different. So indels with a length which is not a multiple of 3 produce a reading frame shift, and all codons after the indel are affected. And so that is really the strongest effect of all of these, say, comparatively local mutations. Now, mutations are a stochastic effect due to the fact that the machinery that replicates DNA is not perfect. And they are due both to the effect of the environment and of the organism internal working. And they can be estimated by comparing orthologs. Orthologs means that, well, you take sequences that code for, say, proteins that are sufficiently similar in different life forms that you can guess that sequences are similar by descent. They have some sort of common ancestor. And then you try to guess how long ago this common ancestor existed, and then you can accumulate, say, you can sort of estimate what is the number of changes that have occurred between one and the other and the common ancestor. So this is what you are going to do, something like that. You have 2 life forms in which you have a comparable sequence in the DNA. One is this sequence here, and then this other one, it's very similar, but is a little bit different. You see here you have a G, here you have a C, here you have a G, and here you have an A. And so you say, well, okay, if these 2 sequences happen to have a common, say, are identical because they have a common ancestor, the common ancestor will likely have the same nucleotide here and here, these ones which have been conserved, but then you don't know what there was at the beginning, up here. So it might have been, for instance, that there was a G here that was conserved on this lineage, but then it changed into a C on this other lineage. Now, a way of estimating that is assume that, say, the mechanism by which mutation happens is reversible in the sense that it is as likely, say, that if we go from here up and then down, there is the same likelihood to have some mutation on going up and then going down, as we'll discuss a moment later. And then you can say, well, okay, the separation between these 2 guys, between these 2 sequences, is 2t, where t is the time since the last common ancestor of the 2 life forms. And then you say, well, I have 2 mutations during this interval of 2t, so there is a rate of 1 mutation per t, whatever t is, in this particular case. So this is a way in which you can roughly estimate the mutation rates. And of course, you can sort of adapt it for the different genes that you are considering. There is a caveat in this approach that we are still considering that these mutations are in fact not selected by, say, in natural selection. So they probably, what you can do is that a good estimation of, say, the background mutation rate is looking at silent or synonymous mutations, provided that the selection is sufficiently weak. Now, there is a second caveat which is even worse. So that is that the comparison between two different life forms evaluates the substitution rates. That is, so what you take a life form and you look at the gene sequence. This gene sequence is by and large the same for all individuals of that species, of that life form. And so what you are saying is that basically that what you're looking at is that all the individuals of that life form have a sequence, have a C in that place instead of a G, and that means that this C has been substituted in the population. And this is not necessarily the same as saying that somewhere a mutation arose and the rate by which substitution takes place is in principle different from the rate in which this mutation becomes. So the rate by which a mutation arises is not necessarily the same as a substitution. And then we have to discuss this point. I will see that when I discuss drift. Because there is an important result that says that if you look at neutral mutations, then the rate of substitution is the same as the rate of mutation. So this is a second caveat you have when you interpret a mutation rate. And now the estimate that usually are done to get at what is the background mutation rate is based on these four general assumptions, and I just want to stress that all these assumptions are false, but of course they can be qualified. So the rates of mutations are uniform. They do not depend on the position of the genome. They assume that they are constant in time. They are the same for the two branches, because you have to consider two branches. And then the equilibrium frequencies of the nucleotides are the same for the ancestral sequence and for the two evolved ones, because you can say that, well, okay, at the beginning, if I had a different frequency, for instance, of G's and C's with respect to the A and T's, and then they tend to change systematically into higher frequency of A's and T's, then of course this way of estimating mutation rate will not work. Okay, so what is the kind of, say, how you describe this nucleotide substitution? What you imagine that you're looking now at a particular site in the sequence, and then you say, well, okay, what is the rate of substitution of, say, of a given nucleotide? Well, or actually, you look at the sequence in a certain region, and then you want to evaluate what is the rate of substitution nucleotide I to nucleotide J, where I and J are, of course, valued in the, say, for the four nucleotides. You look at the frequency in the sequence of nucleotide I, and I define this as F, I, or T, and then you can write down a master equation for this frequency that is simply given, well, if you're given this rate of substitution I to J, the frequency of nucleotide I will change because a J mutates into an I, and this will increase it, or an I mutates into a J, and this will decrease it. And so this is just a classic master equation for this substitution within the four nucleotides. Now you solve for the equilibrium frequencies by assuming, say, by setting this derivative to 0, and you assume that in fact the evolution is such, at least for the stretches of DNA that you are considering, that you can that the nucleotide frequency are at the equilibrium level, and then you write down an evolution matrix. Evolution matrix is the conditional probability to find the nucleotide J time I instead of a nucleotide I at time t, given the nucleotide I was in that position at time t equals 0. So here I and J, so J can also be equal to I. This is what you want to have, but this is not accessible to you because we don't know what the ancestor is. So what you can have is a divergence matrix. A divergence matrix compares the two the two sequences in two different life forms and say that you, by some means, you guess what is the time which has gone since the last common ancestor. And then what you say is that what is the joint probability distribution that you find I in one form and J in the other form, given that, say, the last common ancestor was T generations ago or T time ago. And this is what you can evaluate. Now, how you can turn that, so this is the equation for the evolution matrix. Again, it's just the master equation that we have written before. And then if you know the evolution matrix, you can write down the divergence matrix. The divergence matrix is obtained by considering the evolution in, say, evolution forward in time on one branch and backward in time for the other branch. So you have the initial condition, which is here. You have this evolution matrix forward for one branch, and you have the transpose of that evolution matrix for the other branch. And of course, at the beginning, what you have is that this xij is given by the frequency of the different nucleotides times the Kronecker delta. Tells you that the two nucleotides are equal, and they appear with a frequency that is equal to the equilibrium frequency of that nucleotide. Now, in the case of the divergence matrix, there is a symmetry. Well, in principle, in an infinite population, you would have a symmetry of the divergence matrix. This is not always true because of sampling errors, inevitable sampling errors. But let's assume it's a small error. Then you have normalization constraints on the diagonal elements. That's trivial because of this. And therefore, the divergence matrix, which is in principle has 16 entries, in fact has only 6 independent parameters. And now, if you look back on how the what is here, in fact, if you assume that, say, you can imagine that if there is some reversibility, that is, if the error on one side is equal to the error on the other side, you can have 6 independent parameters for the 6 different transitions. Okay, so in fact, let's say typically there is a very simple Jukes counter model in which all substitutions are equally probable, and let's see what you can get out. So you have, in this model, you assume that all the non-trivial substitutions have the same frequency. Then, of course, in this case, you have that the equilibrium distribution is flat, and the evolution matrix is given by this formula here, in which you find you start from 1 and you go towards 1/4. And then what is the probability of finding the same nucleotide in 2 sequences given by this form here, in which you have, you see, you have 1/4 asymptotically, and you have a time-dependent quantity here, and therefore, out of that, you estimate this product of mutation rate times to the common ancestor in terms of this probability of observing the same nucleotide in the 2 sequences in this way. So this is a way in which you can estimate mutation rates. In general model, you have 6 parameters, and because you have a substitution A goes to C implies the corresponding substitution T going to G in the opposite strand. So this is why you have only 6 parameters. But in fact, if you care, well, okay, that's probably too much. In fact, you find that there are only 5 independent quantities if you only have synchronical data, because of the Sharp-Graph rule that tells you that there are as many A's as T's, as many G's and C's. And so you have, say, there is one parameter that cannot be identified. Now, in terms of modeling, it makes sense to use some simple modeling which allow you to, say, a sort of tame tame the effects of mutations. We often want to model mutations in, say, you are given a wild type, and then you just start looking at what are the effects of mutation. One possibility is just imagine that each mutation produces a completely new genotype. This is called the infinite allele model. And in this case, you have no structure in the mutants. Each of them is as different from the others as from each other and from the wild type. The other one is infinite site model. So the idea is that each mutation hits a different site. In this case, what you are imposing a hierarchy on the mutations. That is, you have a wild type with 0 mutation, then you have one, say, all sorts of different mutants, but with only one mutation, and then you have mutants with 2 mutations and so on. So this is, since this infinite allele model, infinite site model you find quite often in the literature, is useful to remember what they are about. So infinite allele is with no structure, infinite site, there is this class, so you have a class structure which counts the number of mutations in each class. Now, let's now put together mutation selection. Let's start with a simple model in which you have only A and B, and we have a selection coefficient, say for instance S, which is the difference between the Fisher fitness of A minus the Fisher fitness of B. In this case, S positive means that A is selected positively, but we have a mutation probability that A goes into B and vice versa. It's like a, in this case, you write down evolution equation for X, which is the fraction of individuals, of A individuals in the population. It will have 2 terms. Say one term is the selection effect, which is, so it is this selection coefficient here times X times 1 minus X. And of course, that tells you that there is a fixed point at X equals 1 and a fixed point at X equals 0. Where selection is no more working. But then we have the effect of mutations. The mutation, you have a positive term when mu times 1 minus X, and it means that a B can mutate into an A, and then there is another term which is a negative term, which is an A mutated into a B minus mu X. And so out of that you get this form here, and what you see is that this is the derivative of X with respect to time. This is X, and where you find a curve that has a fixed point here somewhere. That means that because of mutations, now you have always what is called a polymorphic population, because the selection and mutation balance. This is called mutation selection equilibrium. We are implicitly considering that the population size is infinite, and we have a very unrealistic model for mutations because it tells us that the mutation just turns one allele into the other and backwards, which is usually quite unlikely. So you can discuss what happens at a fixed point with different, okay, that's not interesting. One thing that you can ask is that, well, we know from the fundamental theorem that if there is a polymorphic population, there is selection is going on without mutation, then we end up with the optimal, and with the optimum among the variants that are present in the population. In this case, in which we have mutation selection balances, what is there some sort of optimality? Well, it turns out that in this case, the fitness is not maximal at the coexistence. But there is something else that is maximal, and say physicists are very fond of this result. Say, define this quantity that has one term is just the average fitness in the population, and then there is another term which is proportional to the mutation rate times this log of x1 minus x. So this looks like a minus energy, and this looks like an entropy, and then you can simply write down the time derivative of this quantity, and you do one line of algebra, and you find that this derivative is always positive unless you are at the fixed point, whichever fixed point it is. And in fact, when x is equal to x star, is this thing here that becomes 0. So this fact that this is always positive tells you that it is optimal at the steady state. Well, you can play the same game with multiple alleles, but then you have to do some games. So introduce some matrices called a Shahshahani matrix, not matrix. And then you have an evolution equation of this kind, and then again, if you assume that there is a very special mutation model, then you find again some sort of optimality. But again, in this case, you have this very, very special mutation model that is unlikely. Now, you will find that so sometimes it's quasi-specious model. The quasi-specious model is a mutation selection model, but in a very large sequence space. So the idea is that you have a sequence which is long. You have a large number of alleles because this sequence can mutate in very different ways. No? Let's assume for simplicity there is a mutation rate, say, of a sequence on a different sequence, which depends on some sort of notion of distance of the different alleles. Can be simply, say, 1 mutation, 2 mutations, 3 mutations. So this is a sequence. K is L are sequences, and there is some sort of mutation rate, QKL, that depends in some structure of these sequences. And then you ask, what is the evolution equation for the composition of the population, in which these 1 to R labels the sequences now? And the population is very large. We are neglected fluctuations. And so you can write down pretty easily the evolution equation. I write down the result, and then I explain a moment. So what you have here, this is the say non-overlapping generations at the so your frequency of a sequence J at time t plus 1. Here you have a frequencies of sequence K at time t. These individuals of sequence K have a fitness WK, so a single individual will produce a WK individuals with the same sequence. But then with the probability K, say QK, JK, this sequence K will mutate into J, all right? And now I sum over all the sequences here, and I get, say, the expected number of individuals of sequence J. In order to get this fraction, I have to divide by the total growth factor of the population. The total growth factor of population is given by the average value of this fitness W. So this is the quasi-specious model. So let's remind what enters a quasi-specious model. The population is infinite. The mutation rate depends in some sense on the distance of alleles. And so you can classify. Of course, that is a sort of, again, strange, because you need to have that the number of individuals on any given species, on any given sequence that you are interested in is large enough so that you are able to neglect fluctuations. But it was discussed a long time, so we go, how to solve, what does this equation tell us? Well, it's easy, the easy thing is to get rid of this this factor here. We just define y, which is proportional to x. So y is an unnormalized frequency factor. And then you have a very simple linear equation, iteration equation like that, in which you can see that these entries here are all non-negative. So these quantities all non-negative, it's a matrix, a TJK with non-negative entries. We can decompose it according to its right eigenvectors. And we can use the Perron-Frobenius theorem, which tells us that the largest eigenvalue is positive and non-degenerate. And therefore, there is a unique right eigenvector psi 0, which belongs to this largest eigenvalue. And therefore, if I iterate this mapping over Y, what I end up with after some time is a vector which is proportional to this unique right eigenvector belonging to the largest eigenvalue. And because now I normalize, and therefore I know that the composition of the population will reach this stable state in which, so as t goes to infinity, what we end up is that the composition will be given independently on the initial condition by this right eigenvector. So that is the solution of the quasi-species model. Now, how is this eigenvector made? Well, let's consider a simple case. We have one optimal genotype, which we call, say, the wild type. And then we have all the other ones have a smaller value of the fitness. So we have, and we have a large number, that's important, we have a large number of sequences. Then we have the mutation probability for going back to the original sequence is very small because the number of sequences is very large. Define W divided by W0 is, so W0 is the fitness of the wild type, and all the others have a smaller value. So it's, say, the W divided by W0 is 1 minus S. And then you can write down simply the equation for the frequency of the wild type. It's given by this expression here, in which I define U is the mutation per site times the length of the sequence. So this is, U is the mutation, is actually the total probability of mutating into any other sequence. And then you have this expression here, and what you see that there is some problem. There is some problem here. We can see it in the next slide, that the fixed point is given by 1 minus U over S. U is the mutation probability, S is the selection factor. When the length of the sequence goes to infinity, you end up with this expression. Of course, when U is larger than S, this becomes negative. It means that, in fact, the only solution you can get is X0 equal to 0. And this is the approximation you can have for different lengths of the sequence. For 10 bases, 30 bases, 100 bases, what you see is practically this guy. So that means that if you have a very long sequence, even if it is selected positively, then at a certain value of the mutation rate, the probability of keeping that sequence in the population vanishes because of the accumulation of errors as the process goes on. And this is known as the error threshold. And in fact, you can look at the number of, say, frequency of individuals in different classes. There is, say, lingering confusion about the error threshold. Say, in the error threshold, we are considering a population of a given size, which is actually infinite size. And we are not considering what happens to the population at large. No? Now, this is different from considering what happens if you have a population in which, for instance, the fitness is so small that it cannot sustain itself. In this case, of course, the population size decreases and eventually the whole population goes to extinction. And in order to discuss this point, let's take a simple model in which we have a wild type with a given right fitness W0, Then we have one mutant with a fitness which is positive, but smaller than W0, and it has one mutation class with the smaller one. And then if there is a further mutation into this mutant, it goes, it's killed. It has fitness 0. So this is a very simple model in which we have this W0, W1, and U0, U1 as parameters. And in fact, And here we have to take into account also the fact that the W0 and W1 are really the expected number of offspring of individuals of class 0, class 1. So it's not only the ratio that is important. So you can write an evolution equation now for the population vector, which is different from the frequency vector. Here, n's are in fact the number of individuals of type 0, type 1, and type 2, and for simplicity, we consider them as real numbers. And then we have an equation like that, which is again a linear equation with a transition matrix. Transition matrix you can see here with non-negative entries, but there are some entries which vanish because of this. For instance, here we see that if we have individuals of type 1 and they mutate by this ratio, then they will simply be killed again. Oh, okay. Now, you solve the problem for the composition, and you look at, again, it's essentially the same mechanism that we had before, but now there is an interpretation to the maximal eigenvalue. So the maximal eigenvalue tells us how the size of the population changes with time. So the number of individuals in the population will be proportional to this maximal eigenvalue times to the power t. And therefore, if this maximal eigenvalue is smaller than 1, the population eventually goes to extinction. On the other hand, the error threshold corresponds to the loss of the wild type in the population. So this condition on the error threshold is given by this one, which tells us that essentially the loss of the whenever this condition this is the condition for W not to be equal to 0. So what you that's probably the best thing is to see it in this way. Here we have a situation in which there is the condition for the error catastrophe that happens. So what we have here is the mutation rate from the wild type to the first mutant. And this is the maximal eigenvalue. So when the maximal eigenvalue goes below 1, there is extinction. And the condition for the error threshold is that essentially the wild type should reproduce, so taking into account its rate of mutation, should reproduce faster than the first mutant. So the error threshold is here when the mutation rate equals, when the mutation rate times the selective advantage of the wild type equals the mutation rated selective, 1 minus the mutation rated selective advantage of the first mutant. And here you see that extinction, so at this point, here we have a population with, say, wild type is present in the population. Here there is error threshold, wild type is lost, but we have a W1. We have the first mutant, and then here we have extinction. So in this case, we see that the error threshold, the error catastrophe, error threshold delays the extinction. On the other hand, if we have a smaller value of the selective smaller value of the fitness for the wild type. So we go to extinction even when we only have the wild type, when the wild type is still present in the population. So we have here. So in this case, the extinction prevents the error catastrophe. So the two things are two separate concepts, and they should not be mixed, and sometimes you find that they get mixed in the, this, if you like, it's very nice to evaluate in the parameter space of mutation rate and the fitness of the wild type, which phases, so this is a unsustainable phase, this is an order phase in which you have a wild type, and this is the disorder phase in which you have only the mutant.
What do you mean by order phase?
Order phase, it just means that you have a wild type, the wild type is present in the population, and you are this side of the error threshold. Okay, that's just a name I gave it. Now, 10 minutes, yeah. So I just started discussing what is drift. Drift is a strange name for us, but it's the one which is used in the literature. I just wanted to mention, say, most of what I'm telling is, was, discussed by Kimura, but it was also contained in a very fundamental work on the early, say, in 1920 by Fisher and Stuart Wright. So this is why Wright is W fitness, S fitness, and Kimura is the guy about for drift. Drift is the effect of sampling fluctuations in a finite population. So you should, in population biology, drift is a random component. It's not what we are accustomed to in physics for the guys who make random processes in physics. Let us consider a simple model. This is Wright-Fisher model. So I had to put them, both of them. You have a population, finite population size, a certain number of individuals of type K, whatever K is, and you have a fitness WK for individuals of type K, non-overlapping generations. And then you are given the composition vector of the population at a given time, so that this is the frequency of type I. And then you ask, what is the number of individuals of type I or K in the next generation? Well, this is just a trivial multinomial sampling problem. So the probability to have a composition n'1 to n'(r) is given by, say, this multinomial coefficient here, times the frequency of, say, times this factor here, which is related to the frequency of this type in the previous generation, but weighted according to its fitness. So what we have here is something that looks, it's really a multinomial sampling, but in which the probability of being chosen is given by the frequency in the previous population times the fitness of that type normalized by the average value of the fitness over the whole population. And in this way, this depends on the actual composition of the population from generation to generation, and it changes in time. But this multinomial can be approximately distributed as a multivariate Gaussian with a certain mean. And so if you look at the, just at the number of individuals of a given type, you can say that this is a Gaussian with a mean which is given to total size of the population times this probability of being chosen. And the variance is just the usual variance when you, say, binomial variance, when you have sampling with a given probability over n, a sample of n samples taken with this probability. This is the variance you obtain. All right, so this is how it looks like. You start with a very colored say, varied population, and then you iterate the process. And because of the fluctuation, you find that eventually there is one type which dominates and goes to fixation. And from then on, as long as mutations are not present, you have just a monomorphic population. Now let's look at one realization of the process. Here I have a population of 1,000 individuals, and here I'm looking over 2,000 generations, and the colors are the different types of individuals you have. And this is very, say, look it carefully. What you have is that the sizes, say, the number of effectives of individuals having different types changes with time, and these boundaries look to do some pretty much an unbiased random walk with a little bit of difference. And once they hit either 1 or 0, so here, no, actually they hit 0 here and here, no, just the number of individuals goes to 0, here it goes to 0, they disappear forever. And the reason is that if we look what we had before, when psi is equal to either 1 or 0, then the variance vanishes, and therefore you have no more fluctuation. But say, important, you have, one of the things is that you have this kind of random walk of these, say, random fluctuation of the sizes, and then what you see is that about, say, about 1,000 generations later, essentially you're left with only 2, 1, and then eventually just one type remains. So this is a different map. So what I'm looking now, it's only one type. Again, it's a population of 1,000 individuals. I start with, I think, a quarter of the individuals have a certain type. I forget about all the rest, and I just look what happens to them as time goes on. And here you see that their number fluctuate, and then sometime go to extinction. Here, you see, they go to extinction. And in this case, they go to fixation. That is, they overwhelm the whole population, and nothing happens after that. And the only one case here, say it was undecided after 2,000 generations. So these are several realizations of the same process, and there is no selection effect. It's just a random sampling. And out of random sampling, what you have is that different populations will end up, say, in some population this goes extinct, in some populations they will go to fixation. So different populations which start with identical initial conditions will have different fate. What happens if we have that one type is selected, positively selected? This is one simulation. Here it's probably not terribly clear, but what you can see is that at the beginning, there are still the same fluctuations somehow, and then, say, at a certain point, it seems that this, say, light blue takes over and starts increasing exponentially. But there is some region here in which the effect is, say, this fluctuating size is still present. And again, I look at several realizations of the same process, and here I have to cheat a little bit because I took a smaller, first of all, I took a smaller population size, and I took a very small selective advantage for the, say, for the type that I'm interested in. And so what you see here, you have a certain number of realization, and in fact, you can find that in 5 cases out of 10, it goes to fixation. In 5 cases out of 10, it goes to extinction, and it takes, in this case, about, say, 1,000 generations to reach a fixed point. So this, I think that this is a good stop. So it is often convenient to consider natural population not so much as an aggregate of living individuals, as an aggregate of gene ratios. such a change of viewpoint similar to that familiar in the theory of gases. Okay. The term drift has become quite popular among biologists. However, in mathematical theory of Brownian motion, the term drift originally connotes directional movement of the particle. Therefore, in our context, the adjective random should be attached to it. So this is, again, mentioning the fact that you have this interpretation of drift. So this is finite population implies different outcomes for different experiments in the same conditions. For the people who work in statistical physics, this is called lack of self-averaging. So necessarily you have to describe an ensemble of population. And we can use the theory of Markov process, and the way of simplifying the mass is using a diffusion equation. This is what I'm going to discuss next time. No, is that? Huh? That's it. Okay. We stop for a while and then, yeah, yeah, good. 10 minutes break, yeah. Badly needed.
Can you go back to the master question, please?
Yeah, but first I would like to
Okay, so questions?
So I think I might ask you this before, but for a non-physicist, what's the intuition for that phi of x that you define as a term which has entropy, you know?
It's a compromise between a selection and the mutation rate. So imagine that your microphone is out, yes. Also the yeah. So it is a compromise. Imagine that you have a strong selection, then you end up with the optimum. Now you turn on the mutations, that means, there's a Larsen effect. You turn on the mutation, and then that means that you are going to have a number of mutating individuals that are less selected, so they probably will leave fewer descendants. And there will be some sort of equilibrium. The population will not made only of the optimal ones, but also say, of a cloud of mutants that are around the optimal one. And in these simple cases, you can find that this cloud is obtained by optimizing some function, but I wouldn't I wouldn't like to press it too much on the interpretation. There is really, because it really depends on first of all, on the fact that you're looking at infinite population limit, and second, on particular mutation rate. And in fact, if you look at what mutation rates are, even at the level of nucleotides, you find that there are some rotations. I'll say that some nucleotides tend to mutate into different nucleotides, and then these different nucleotides mutate into different nucleotides and then goes back. So there is a curl going on. And that this optimum does not work if you have a curve.
Right before the entropy and energy analogy, the problem was that the average fitness did not reach a maximum at the x of equilibrium, right?
Sorry?
The average fitness did not reach a maximum at x at equilibrium.
Yes.
Biologically speaking, does that mean that the mutation rate does not allow for the fitness to reach a maximum at the whole population? Is that it?
Yeah, but if you have mutations, you simply cannot have a monomorphic population because you have the mutants. That's as trivial as that. Now people have been speculating whether there is an advantage, for instance, there has been a situation in which you allow a number of mutations that do not change too much your fitness. That is called the selection of the flattest. But in some cases, you see that if you look at the selection for the, say, what Curtis was talking about, a selection for the sequences, for the binding sequences of transcription factors. There is definitely a sort of entropy component into that. So some sequences are, probably evolved in such a way as to buffer, that is the word that people use, to buffer mutations, keeping functionality.
Yes. So in the quasi-species model, one of the assumptions is that mutation rates depend on the distance of alleles. And what is that distance of alleles, and what would be a biological interpretation of that?
Well, the quasi-species model, the idea is simply really what happens if you have a great deal of possible alleles. And this is where the thing comes in. And then you can simplify whatever. So the basic idea is that instead of looking at, like in the first model that I have considered, the probability of reversing the mutation is high, What happens if the probability of reversing mutation is very small, then you're likely to lose the wild type. That is what the problem is about that. That's all the motivation. I wouldn't say go too much into that, because that's the message. The message is that usually what you have is that you have a very well adapted, in principle, adapted sequence, and it is easy to lose it. And then you cross the error threshold.
I have a question. Can you comment on this eigen paradox that you cannot have a sequence, you cannot evolve a sequence that is large enough to correct for mistakes because you can never get there, because for short sequences you have this threshold?
Yeah, okay. That is the interest of the say, error threshold. So the idea...
Could you restate the paradox that you're going to explain?
Yeah, I'm going to explain it. So the idea is that imagine that you have a sequence of a certain length L, and you have a mutation probability per site. And then you ask, what is the probability that a wild-type sequence mutate into some, say, what is the probability of keeping the sequence with respect to mutation? So this probability of keeping the sequence will be something like 1 minus mutation probability to the power L. Now, the whole idea of the error threshold is that the loss of the number of individuals of the wild type to the mutants, which is due to this probability of mutation, should be compensated by the reproductive advantage of the sequence. So if you put these things together, that you have that the advantage must be larger than something that increases exponentially with the length of the sequence. And therefore, you have that, say, any reasonable, how to say, the advantage should be, how should I put it? The length of, given a certain advantage, the maximum length that you can tolerate without making mistakes is bounded. And if you put inside the numbers, you get that it will be of the order of a few hundred, well, 100 and something nucleotides, and that is not long enough to make, say, a working replicating ribosome, because the probability of mutating increases with the length exponentially. So that is the paradox. A way of solving the paradox has been suggested, say, basically what you want to have is that you have a self-replicating molecule that collaborate in order to perform the replication, in order to correct one the other, say. So there are several mechanisms. Eigen imagined that it was possible to have a situation called the hypercycle, in which you had several self-replicating molecules with errors, but they each collaborate in correcting their errors altogether. But this is a very unstable situation. And then the way in which people like Maynard Smith and SMRI suggested was instead that considering effects of group selection, that is in some sense similar to what, so the idea is that if you put self-replicating molecules together that are able to compensate for their mistakes, then it is possible that the group formed by these molecules is able to self-replicate even in the presence of this error. But, okay. I'm not. Yeah.
Luca, in the quasi-species model, it's clear that this delay is very strange. It's clear that mutation is linked to reproduction or replication because it happens as a replication error.
Yeah.
Well, that's very clear. Yeah. But in a simple model, the first model that we studied, I cannot see that. It seems that mutation is an independent process of replication.
Yeah.
You see, it appears as a sum in the equation. Could you comment on that? Are there different models or?
Well, say even in the quasi-species model, you can. I made a quasi-species model of with non-overlapping generations, and usually with non-overlapping generations, it's easier to put a mutation up, say, as a step before the replication. In continuous time, it's easier to work in saying, well, okay, I have evolution in which there is what is called a parallel mutation selection model. And you can also do that, of course, in discrete time, but that is a sort of more awkward. But in continuous time, it's just easier to say, well, okay, either it mutates or it reproduces. I agree with that. You hope that these details do not change much of what you're going to get. Proper formal helps. Yeah.
So in both scenarios where we have infinite populations and finite populations, the result is sort of on the spirit of the strongest dominates very rapidly, right?
If you have a small population?
No, in both cases, let's say, one, you stated the result of the Frobenius theorem, where the strongest allele will dominate. And in the finite model, you also showed us in some simulations that after not many generations, you had dominance of the strongest allele. My question is, you know, either in nature, there is not such a strong competition that something would dominate, or maybe what are your feelings or why one sees several alleles coexisting?
Yeah, okay. So this is a problem. What we see is that there is variability in a natural population, and then one of the thing that you can infer is that most of this variability has very little, if any, effect on fitness. This is something that you can, not everybody agrees. We do know that there are some variabilities that are related to fitness effects, but they are still there. I think that there is also a great deal of variability, for instance, in human population, which has effects on fitness, but is at such a lower level of frequency that it doesn't really change much. And this I'm going to discuss next time, the fact that even if an individual, say, even if a variant has higher fitness, that does not mean that it is bound to dominate the whole population because of the intrinsic randomness of the sampling. So this is what we are going to discuss. So you know, and so that means that there is a certain amount of variability which is also related to the, say, which has definitely a fitness effect that is tolerated, and this effect also raw material for evolution.
Professor, among undergraduate students, it's common to think using adaptationist thinking, you know? Mm-hmm. Selection is the most important process explaining evolution, for example. This is really common among undergraduate students. So how relevant drift could be to the evolutionary process? Could you clarify to us, please?
So, again, adaptation thinking is saying that whatever happens happens because it is selected. I think that this is really too narrow, because the real, as I mentioned, the very important problem is that you have variability. Variability is the raw material of evolution, and it is kept. So all natural populations have a degree of variability, which is where evolution is acting upon. So just considering the effect, in fact, it is much more interesting to see those cases in which adaptation is not perfect, because they tell you something about the evolutionary process itself. So this, say, strange paradoxes in our anatomy, and I don't know if you have heard of the idea of the recurrent laryngeal nerve. Have you heard that? Can probably, if I may. Okay. This is, okay. So this, the yellow guy is vagus nerve, which connects essentially thorax to the cerebellum. And out of that, you have a recurrent, recurrent nerve that go in the opposite direction. One is this is the right one, it goes directly to the larynx. And then there is the left one, which goes all the way down under the aortic arch. And then it goes back to the larynx, or to the bottom part of the larynx. So it makes this deviation of several tens of centimeters in order to say across here is less than half a foot, but then it goes all the way down. Now this is probably not so special for us. I mean, it's it looks strange, but just look at what happens to the giraffe. A giraffe has 4.6 meters of this nerve. It goes from here to there. It goes all the way down and then up again. And these are, and say, what is the reason? And this is the accepted reason. Say, if you look at the fish, the structure of a fish you see is much more regular. You have, say, nerves on the one side, and so these are the nerves, and these are the blood vessels around the gills. And so you just look at what is the whole anatomy has been changed, but here we have this fourth vessel here. This will become the aortic arch, and this is the predecessor of the vagus nerve, and then this guy had remained stuck under the aortic arch and could not cross it. And of course, it makes more sense to make a longer nerve than making a longer aorta.
And last question.
I don't know if it's a question. There are people who do experiments that have something to do with evolution, but they do it on RNA. Like Jerry Joyce has he sort of spontaneously evolves self-reproducing RNA molecules that do so in an autocatalytic fashion, and then Jack Szostak has some similar sort of experiments. Does one learn the same lesson about the mathematics of evolution from those experiments, or is it often some totally different branch?
Well, the problem there is that it is still a very contrived system. It doesn't seem to open a vista for further evolution. You have to be in a very carefully chosen setup, and then it works for, say, but I don't know if it will open, say, for further improvement. What you need is something that it is more robust, probably, and more open to development, and that's what they get. But this is just the beginning. I mean, it's a wonderful work. But I'm say so far they reproduced some of the mechanisms, but it had to be very contrived in order to be able to do that. On the other hand, for instance, because it is simpler, people who have done evolution of populations of computer programs, there you have open-ended evolution. And you have to say, to kill the experiment at a certain point, because they had something coming out that was completely not anticipated.
Do you ever see an evolutionary shift, as it were, in the computer program evolution? That is to say, the coming together of, so to say, subunits to form a self-reproducing higher order?
This has been observed. This has been observed. And it is, say, parallel to something that has been observed in viral population. But say, again, it's intriguing. So in some viral populations, this is an actual experiment. They infected, well, it was not with a phage. It was, unfortunately, with viruses on mammal cells, but with high multiplicity of infection. And they developed a kind of viruses that could only infect if they were, or two kinds, if they were both present in the infection. Because of course you have an advantage, each of them reproduces faster, but then you have a disadvantage that it is, I mean, it would be crazy to do that in the wild, because in the wild, if a virus succeeds in getting a target, it's very lucky. But you can have this kind of, say, symbiosis. And the analogous thing was seen in the program. It just arose.
Okay, I think this is a good point to stop. And we thank Luca again, and we resume at 2:30.